story, and if you want to look at the full story, you can look at this QR code here. And I thank the organizer for the invitation, and thank you, Jen.
Thank you, David. You can stop. Oh, no, actually, don't stop your screen sharing just yet. If anyone has any questions, please post them in the Q&A box. So, David, the first question is, what was the light intensity when you compared photosystem 2 and the photosystem 1 protein quantification? If there is high light, there is a possibility that PS2 content may decrease in the SEPA1 mutant.
Yeah, that was just, we just tried the standard light conditions, so 150 microeinsteins. So there was no, we didn't subject the mutants to any challenging highlights.
Okay. Another question that we have by Alizé Malone. There were 3 bands for SEPA1. Can you say a bit more? Do you think there are trunculated products and relevant for function?
So yeah, we think that it's relevant. So I didn't have time to mention this, of course, but we think that they are just fragments of SEPA1. And what we think is happening is that in the second dimension, you can see the level of SEPA1, the mature protein, and you can still see these fragments, but they're only present in the monomeric fraction. So what we think is that SEPA1, once its function is fulfilled, it's getting cleaved by probably proteases, and then you still find the monomeric fraction, but only you find these fragments, but not associating with complexes anymore, but just in the monomeric fraction. So that's why we think that it's a result of basically the end of a SEPA1 function.
Okay, thank you very much. I think that's good for now. Thank you again for a great, wonderful talk. And now we will move to our last speaker, Clara Groth-Krago. Clara recently completed her PhD in plant evolutionary genomics at the University of Vienna in Austria. And currently she's a postdoctoral researcher at the group of Ovidiu Paun in Vienna. And Camille, sorry, I'm not sure how to pronounce his name, Camille Roux in Lille in France. Her work is focused on understanding the evolutionary forces that drive adaptive radiations. She will present her PhD thesis research, for which she used genomic data to disentangle the molecular sources of variation underlying adaptive radiation in Tillandsoid bromeliads, which is a neotropical plant from the Americas. In this work, Laura especially focused on the evolution of CAM photosynthesis as a key innovation trait and driver of diversification in the subgenus of Tillandsia. Today she's going to speak about how CAM evolution is associated with gene family expansion in an explosive bromeliad radiation. So Clara, the floor is yours, and good luck.
Thank you so much.
Please share your screen.
Yeah.
Yes.
Let me see. Can you see my screen now?
Yep, we can see your screen. You're good.
Okay, perfect. So thank you so much for inviting me today to give a talk. I'm really excited about this. And my name is Clara Groth, as Guy already introduced me. My talk is quite a bit different from the 2 previous talks we've seen. I hope it's interesting for the audience. And it's all about CAM evolution in this bromeliad radiation and how it's associated with gene family expansion. So I want to start by introducing the plants that I worked with during my PhD. This is the subgenus Tillandsia. It's a clade of about 250 species that belong to the Bromeliaceae family, or also known as the pineapple family. And they are considered an adaptive radiation because they diversified very rapidly in the last 2 to 3 million years into hundreds of species. And it's not really well understood how this really can happen at such rapid rates. So that's a very interesting topic for evolutionary biologists in general. And this diversity goes hand in hand with also significant ecological and phenotypic diversity. So in the subgenus Tillandsia, we actually especially are interested in a certain number of key innervation traits. These are traits that have evolved often repeatedly in the group and that are associated with diversification. Examples are the epiphytic lifestyle, so these plants grow on other trees. For example, you also have specific evolved types of trichomes as well, a very large variety of flower color and structure. But what we also have as a listed key innovation trait in this group is the evolution of CAM photosynthesis. So I will actually introduce CAM a little bit later in the talk to catch everybody up on that. But it's repeated independently over many plant families, and because interesting trait for evolutionary biologists because it seems a complex trait, but it has evolved so many different times, making it paradoxical. So that's also another reason why we're interested in looking at this. Subgenus are just interesting from a so that it was not quite dynamic.
Recording in progress.
Radiation generally. And so this made us wonder, is this dynamic genome evolution in any way associated with the ecological diversification that we see in this group? And could we perhaps think that this genome evolution might be underlying this diversification or the evolution of these key innovation traits? And so that's what this project tried to address in some kind of way. We wanted to first of all understand better the genomes of these plants, but we also wanted to see whether differences, diversity in the genomes between plants could be linked back, especially to CAM photosynthesis. So to do this, we selected 2 species which represent the phenotypic extremes, let's say, of the photosynthetic metabolisms in the group. Starting with Tillandsia antiquata, there's a strong constitutive CAM plant, and then Tillandsia leibowniana, which at the time when this project was started, based on carbon isotope ratios, was indicated to be a strong C3 or like a full C3 plant. And the two are quite closely related within the subgenus, so they also underlie a shift in photosynthetic housing. So what we did was to get PacBio data, Hi-C data, and Illumina sequencing data, and with that create reference genomes for both species. And here I just want to show an overview of these 2 genomes that we generated. They are relatively contiguous, so we were able to obtain about 80% of the reference genome in 25, 26 main scaffolds, which is also the approximate number of haplochromosomes we are expecting in these plants. And these large scaffolds
Recording in progress.
which is higher in leibowniana. You can also see that here you see the per scaffold repetitive content in yellow, and you can see that a really big chunk of the scaffold, whereas in fasciculata this is less pronounced. So with these reference genomes then, we wanted to actually compare them and understand how these the genomes of these species are different. So we started by looking at their karyotypes, again with cytogenetic work in this case, and we found that actually there is a significant change in karyotype between the 2 species. in that Tillandsia laeboldiana has 6 chromosome pairs than Tillandsia fasciculata, and also then the majority of Bromeliaceae, which is quite significant for 2 species that diverged for the last 2 million years. And as you can see, they also seem larger, these chromosomes, so we started to think that perhaps we have had cases of chromosomal fusion that may explain this difference in karyotype. So that made us really interested in understanding large-scale rearrangements and whether we could define these in our genomes. And so for that, we performed synteny analyses, and for that, we also added to the mix pineapple, which is about 50 million years diverged from these two, and one of the only few other reference genomes available for Bromeliaceae. And as you can see, the synteny, which is one single orthodox and reflects sort of how similar the organization of the genome is between the species, you can see that this is quite well maintained between pineapple and fasciculata, but however, between the two Tillandsias, you see a whole lot of reshuffling, which is, of course, again, quite surprising. We were also able to describe 1 chromosomal fusion, or what seems like a chromosomal fusion, and 2 reciprocal translocations. And then we wanted to ask whether these rearranged scaffolds had a significantly different selection landscape from the non-rearranged ones. And we did that by calculating the DNDS ratios of copy gene orthodox on all of these scaffolds. And you see here the distribution of DNDS for, in this case, scaffold 14, compared to all the other non-rearranged scaffolds in Tillandsia laeboldiana. And we couldn't really see much of a shift, so it doesn't seem like these rearrangements have really affected the DNDS ratio. So on that time scale seems to not be too different. And the reason why we were thinking this might be different is because rearrangements in chromosomes can have an impact on linkage disequilibrium, which then has an impact on selection as well. And based on Marie-Laure de la Harpe's work, we were very interested in gene family evolution as well, and we did see that the rate of gene family evolution was different between the 2 species, because fasciculata, the CAM plant, tends to have about 20% more multi-copy gene families than laeboldiana, and also these are larger in general than in laeboldiana. But so far, I've not really linked anything back to CAM photosynthesis. I've just been describing how these genomes are similar or different from each other. And for that, I want to explain to you how we address CAM photosynthesis first. And that's first by explaining how it exactly works. So the main difference between a CAM plant and a C3 plant, which is C3 really, is that CAM has shifted the CO2 absorption to the nighttime compared to C3 that is during the day, and they immediately feed CO2 to the chloroplast and start photosynthesis just in that way. In CAM, on the other hand, CO2 is converted to malate and stored in the vacuole, and then during the day it will be reconverted to CO2, and that allows the plant to keep its domain closed, which makes these plants generally also more water use efficient, and is one of the reasons why CAM is often associated with arid and hot environments. And so as you can see, this is mostly actually a shift of temporal activity of enzymes. It's not that it really needs novel enzymes or anything like that. So we're interested in describing the CAM syndrome, let's say, in these plants by looking at differences in temporal expression of genes between the two, and also obtaining metabolomic data over a 24-hour cycle. So that's what we did. We placed our plants in a climate chamber, let them acclimate, and then we sampled them 6 times in a 24-hour cycle. And we obtained metabolomic data from these samples and also RNA-seq data. So the metabolomic data is shown here as a PCA, where we just put all of the compounds that were measured to see whether that already differentiates the 2 species. And you can see that the PCA already has quite a bit of a split between the 2 species, just by looking at all compounds combined. And then we were interested to see how malate was accumulating over time in a 24-hour cycle, because that's a typical feature of a CAM plant. It will accumulate malate as CO2 is converted and stored in the vacuole. And that's exactly what you see. You see a differential accumulation curve for the 2 species, where Tillandsia laeboldiana doesn't really seem to significantly accumulate malate, over time, whereas fasciculata does show this clearly, showing us that they are for sure different photosynthetic phenotypes. Our differential gene expression analysis found 911 genes differentially expressed between species in a 24-hour cycle, and this included 68 previously described CAM genes. So now we have a subset of candidate genes that are linked to CAM evolution, and we wanted to know if these genes in particular were characterized by some of the genome diversity or variation that we had already seen in the two plants. But for at least the first few that I'm showing here, we couldn't really find a clear signal. So in terms of, for example, TE insertion in or near these genes, particularly elevated or lower TE insertion than other genes, we do find that the CAM prime tends to have more often a TE insertion in these CAM genes than Tillandsia leibowdiana, but it's only a small signal. We also didn't really find any candidates of the NDS genes, so genes that might have been under selection between the two species that were related to CAM. We did find candidates in other pathways. And we also didn't find that they were specifically associated with rearranged scaffolds either. But on the other hand, we did find an association between CAM-related gene families and that they were expanded. More often, they were expanded in Tillandsia fasciculata. So here I'm showing enriched gotones in our differentially expressed genes that are in some kind of way related to CAM photosynthesis. And these bars are showing whether the underlying genes belong to gene families that are more often expanded in fasciculata. That's when the bar are shifting to the right, or when they are more expanded in leibowdiana. So meaning more copies in one species or in the other. And you can see that there's quite a few of these functions where there is a shift towards gene family expansion in fasciculata. And on top of that, we also described our genes in terms of what kind of orthogroup they belong to, whether they are in one-to-one orthogroups or in expanded orthogroups in the CAM plant, etc. And we found that in our CAM-related DE genes, we have about 3 times more often gene families that are expanded in fasciculata. And we also see an enriched amount of gene families that are multi-copy, but they are equal in size, so their gene duplication is likely to precede the split of the two species. So with that, I want to conclude this work. I want to say that we found quite a bit of divergence in terms of the genomes of the two Tillandsias, even though they are quite closely related. We found karyotype changes, differential accumulation of TEs, differential gene family evolution, etc. But what we really found linked or associated with CAM photosynthesis was gene family evolution. And on top of that, we are not yet completely finding clear signals that TEs or large-scale rearrangements or cloning sequence evolution are really associated with CAM. So I want to thank everyone involved in this project, the co-authors of this paper, field collaborators, botanical gardens, and you for listening, of course, and I'll be happy to take some questions now.
Thank you very much, Clara. Just to remind everyone, if you have questions, please post them in the questions and answers box. And also to the previous speakers, you can go through the previous questions and you can answer them with writing, because we didn't have time to answer all the questions. And so first question, Clara, by Maxim. Great talk. Have you looked at animal feeding behaviors of CAM plants? Would animals feed on CAM plants in the evening when starch levels are higher? A bit of a different question. Apologize. It is an interesting question, though.
Yeah, that's super interesting, but I have not looked at this. I don't have knowledge about this, actually. But it's a cool question. I don't know much about herbivory and Tillandsias, so I'm sorry.
That's all right. And Hugo Lima Melo says he's curious about the next step of the project, if you can describe them a bit. Like if you have a plan, what you're going to do next.
We're currently working on another project that sort of sprung out of this, where we looked at 2 Tillandsia species again, Leibowdiana one of them, and then a different one. And we drought stress them, because one thing about CAM is that it's a relatively plastic trait, and it can happen that a plant that performs C3 in normal or like optimal conditions, if it's drought stress, it will activate CAM. And this is something that hasn't been explored a whole lot in Tillandsia, because you need to do drought experiments, etc. So we did this, and we found that indeed, actually, Leibowdiana can activate CAM. This is also something we already were suspecting from our findings in this work. And then we looked at whether when it was activated CAM, how much overlap was there in the genes that were differentially expressed to the constitutive CAM plant. And we actually found not very much overlap. They seem to be employing different genes even to achieve a similar phenotype. So that's stuff that we're trying to write up now and get published. Yeah.
Well, okay, that's great, fascinating. And last question, I think we have time for one more. You mentioned the expansion of the ATP synthase family. Can you give more details about what subunits? Are they chloroplastic or are they mitochondrial?
Yeah. I was worried I would get questions like this in this seminar, and I remember looking at this back then, but right now I can't really tell you off the top of my head. But it's in the paper. It's really interesting though. The VATPases seem to have expanded quite a bit, and it also seems like different gene copies were being used differently by the two species, so this is definitely stuff to look into more. Yeah.
Okay, thank you very much. It was a great talk and a great study. I think we're done. Thank you to all of our speakers, and thank you to all of you that attended. And I don't know, Mary, do you want to say a word?
Thank you to the editors of the Focus issue as well, and thank you particularly to Guy for the great job you did of moderating the questions. We'll be posting this, as I said, to YouTube. In December, we'll be hosting a plant physiology Focus issue webinar on the topic of root development, so the other end of the plant. Thank you all very much, and have a good rest of your day. Bye.